So if I get distracted, it's because I have the sun setting into the sea. Hopefully it'll be okay. So I'm here to talk about the Transcriptome Atlases of the Craniofacial Sutures project. This is being run by Mimi Jabs, myself, and Han Van Barkel. And Meng Wu is also here in front as a postdoc that'll be on this project. We also work with Michael Donovan at Mount Sinai, and Stephen Potter will be familiar to people from FaceBase 1 who worked on a similar project for early craniofacial development. So the overall goal of the project is to generate comprehensive gene expression atlases for, we've chosen 11 major craniofacial sutures at 2, and in some cases 3, or one case 3, embryonic stages of the wild-type mouse and 2 craniosynostosis mouse models. So the significance of this is that suture function, if you can picture Shannon's great photos of zebrafish mutants earlier, suture function is critical for normal craniofacial development. So when you have defective or pathologic activity of suture genes, you end up with such dysmorphologies as craniosynostosis and mid-face hyperplasia. And apart from, say, syndromic cases, in the majority of sporadic cases, the genetic cause in most of these cases is not known. So our thesis is that a complete knowledge of suture gene expression would further our understanding of suture function, craniofacial development, and aid in predicting genes involved in craniosynostosis and other craniofacial dysplasias. So the specific aims, we have 3. Aim 1 is basically the material collection aim, and aim 2 and aim 3 are data generation. So in aim 1, we just want to isolate the subregions, and I'll expand on subregions later, of 11 murine craniofacial sutures. We're using laser capture microdissection to do this. If you could attempt to do this manually, please tell me how, because I think this is the only way you could do it cleanly. And as I said, 2, and in one case, 3 embryonic stages. And in aim 2 then, we want to use next generation sequencing to generate RNA-seq files for, in the case of aim 2, wild type mice. In this case, we're using Black 6 as our model. And in aim 3, again, generating RNA-seq files for APIT and Sutures mouse mutant models. In this case, FGF receptor 2, S252W, which is an activated FGF receptor 2 mutant, and Sutures, which are mouse twist1 knockout. And these are on the same Black 6 backgrounds. They've been bred in the Jabs lab for many years on Black 6. Obviously, the mouse is a good model for humans. As you can see with this color coordination, the bones are highly homologous between the mouse and the human. And so therefore, the sutures between them, I'm just pointing out, say, cranial sutures, sagittal, coronal, around the front here, or the interfrontal, or as it's called in human, the topic. Joan Rixmeyer has been acknowledged a few times, so I'll just point out that this is a photo from her. So gene expression distinguishes the subregions of sutures. So these are lower magnification and higher magnification of the mouse coronal suture, and we're showing twist1 expression that's restricted to the suture mesenchyme, and complementary to that, FGF receptor 2, which is expressed in the osteogenic fronts. And these 2 genes are at the heart of a model of suture formation, the practical outcome of which is osteogenic expression or osteogenic program, as exemplified by the expression of alkaline phosphatase A, is restricted to the bone-forming areas and excluded from the suture mesenchyme. And gene expression also varies over time within a suture and also over different sutures. So in the case of twist1, going from E14.5 to E18, its intensity and actual sort of location within suture changes. If you look at different sutures, thrombospondan 1 is expressed in the middle of the sagittal suture mesenchyme, but not, say, in the coronal. GLI3 is expressed in the lambdoid and the interfrontal sutures, and in GLI3 mutant mice, these are the 2 sutures that fuse. And the problem, though, with a lot of this kind of data is that people focus on their favorite sutures, or they'll choose a few sutures, but you'll end up not knowing what the expression is in the rest of the craniofacial complex. And there's new genes to be discovered. We know that because we found one. So in the case of BCL11B, this is already known to be expressed in brain thymus development. We found this serendipitously when we were using it as a brain mark, and then just realized it's expressed in sutures. And soon after that time, a paper came out expressly saying that in the BCL11B mutants have mid-facial hyperplasia, but there's no discussion of the expression pattern, and there's no previous discussion of expression patterns in the literature that would particularly relate to this kind of phenotype. So it wasn't a direct goal for our FaceBase project, but it's very relevant to it. So we spent a while characterizing the expression of this gene, basically between E14.5 to E18.5, and throughout the entire craniofacial complex. And this, I'll just use this picture of E16.5 to summarize the results, which is that BCL11B is expressed in suture mesenchyme. So I'm showing you the frontonasal suture, nasopremaxillary suture, maxillary premax, or the maxillary palatine sutures. And if you go from E14.5 to E18.5, the expression is increasingly restricted to the suture mesenchyme. And often the overlap, either there's no overlap between RUNX2 and BCL11B, or there may be like 1 or 2 cell overlaps. And BCL11B is interesting not only because there is a phenotype, and there's probably going to be papers coming out on that soon from other labs, but I said twist and FGF receptor 2 are at this core of a model about how bone formation is controlled at the suture and how you maintain suture mesenchyme. It involves twist homodimers versus heterodimers controlling FGF receptor 2 expression. There are many other factors involved, BMP and IDS being one of them. ID is a gene whose expression is restricted to the osteogenic fronts. And in other cellular contexts, but not in osteoblasts, because it hasn't been known, but BCL11B interacts with all of either these pathways or directly with expression of IDS. So not only BCL11B, but other genes that could be found would just add to our knowledge and our understanding of what's happening in the suture mesenchyme and the osteogenic fronts. So to move on to the specific aims, like I said, specific aim 1 is really just the material collection phase where we're using laser capture microdissection to microdissect sutures. The sutures we've chosen, there's 5 calvarial sutures. I guess I'm using the word cranial here. I'm also including intranasal on this picture, and even though I'm saying E16 and 18.5, I'm showing you E17.5 as a picture, but that's because we don't have E18.5 in this kind of micro CT images. But you can see it's making the point that the sutures are changing in their morphology as they approach each other during these 2 ages. And the facial sutures are the sutures between the maxillary, premaxillary, maxillary, and palatine bones. And almost all of these sutures can be found fused in differing syndromes or sporadic craniosynostosis. And then these sutures also differ in a number of ways that may affect or could be influenced by gene expression or reflect differences in gene expression. So all of the facial bones and sutures are derived from the neural crest. In the calvaria, the bones and the sutures themselves could be either neural crest, mesoderm, or of mixed lineage. You have bone identities that are different. So you have either paired bones that are, paired frontal bones. Just click that off. So some bones are homologous, in which case you may not expect to see differences in expression, and we're not actually going to be looking between homologous bones for differences. But other sutures have non-homologous bones. So in the coronal suture, here you have frontal versus the parietal bone, etc. Sutures also differ in the morphology of the bone. So you have overlapping sutures. This is the palatine maxillary suture. Also the coronal suture is overlapping, but on a very different scale. Then you have bones that meet end to end, but they have very different shapes, such as a narrow area of confrontation in terms of palatine bones, or in the interpremaxillary you have a very wide overlap, a very wide apposition of the bones. So the versatility of these gene expression atlases, we hope, are certainly that you'll be able to discover gene differences between individual sutures. And we're also hoping that'll be a way of discovering differences between lineages, say mesoderm versus neural crest, or between individual bones. And there are papers, say from the Longacre lab, that characterize the differences, at least in gene expression levels, between frontal and parietal bones. Perhaps structure, and then also the timing, so the evolutionary age of the suture. So I'll just describe the method of laser capture microdissection that we're doing. So it starts with just sectioning fresh frozen embryonic heads in our cryostat. Generally, we make a series of slides that cover the entire suture, and then 1 in every 5 or 1 in every 10 slides will go to a regular glass slide. That'll act as a guide slide that I'll describe. And these membranes are plastic covered. So there's a special slide that's required for laser capture microdissection. So we use these guide slides. We stain these with alkaline phosphatase to identify the osteogenic fronts versus suture mesenchyme. And this just gives us a way of like starting to be sure where our osteogenic fronts are. Some sutures are much easier than others to do this with anyway. Then we take our membrane slides. We go to our laser D6500. And basically you sit with your slides in the machine and you can sit at the computer and draw out the area that you want to dissect. So like I said, we have our...
Speak for yourself. You're right, Rich.
So we start with this as our guide, and then when we're at the machine, we can further identify our osteogenic fronts from our suture mesenchyme, and these are dissected. And then there's RNA extraction, and then these are just checked on the bioanalyzer for the RIN score, so the RNA quality and the concentrations. Oh, sorry. So on to the data generation part. So we're in the middle of this at the moment. So the idea for the wild-type sutures is that we're looking at 3 different ages, 14.5, 16.5, 18.5. There's 11 sutures that I've listed down here by their abbreviation. At E16.5 and 18.5, we're looking at all of these. Each of these squares is like one mouse, one suture. And then the diagonal bars represent either the suture mesenchyme that'll be dissected or the osteogenic fronts. For homologous bones, these will be combined, and for non-homologous bones, there'll be separate RNA-seq files. And for the coronal suture, we're just doing that only at E14.5. The coronal suture forms very early in twist and apit mice. It also fuses, and in humans, it also starts to fuse very early, so it's of interest to us. And the workflow is basically the suture microdissection, RNA extraction using the PicoPure kit, and then we're using Ovation, RNA amplification, then to make the TrueSeq libraries for Illumina sequencing. So at the moment, we're just in this stage for these sutures. The goal for our first year is producing these and the twist mouse, the equivalent interfrontal sutures, which I'll get to. Oops. Okay, so on to AIM3, which is pretty much doing the same thing for our 2 mouse models. The mouse models we've chosen are the APIT, FGF receptor 2 activated mutation for mouse model. This is like the most common APIT mutation. The mouse reflects the human, or the mouse phenotype reflects the human, which is embryonic coronal suture fusion to start with. Their interfrontals are wider at P0. There's extensive maxillary, premaxillary, and other facial fusions at P0. And if you have conditional mutants that can survive, you see increasing mutation or fusion of other sutures in the face. FGF receptor 2 is extensively expressed in osteogenic fronts in all the bones, I guess, that have been looked at. And as I said, all sutures will be assessed at 16.5 and 18.5, and the coronal at E14.5. The Sutures, so as I said, I said that earlier, there's this model where twist 1 and FGF receptor 2 are like the heart of controlling suture formation. So we're also using the twist 1 mouse as a complementary model. We also have coronal suture fusion early and wider interfrontal sutures. Twist 1 also interacts, as I've said, with FGF receptor 2 and other pathways in suture fusion. And we'll just be looking very limited number of sutures. It's the coronal and interfrontals at E16.5 and E18.5, and the coronal also at E14.5. And these tables just show that again for the APIT. We're pretty much doing exactly the same sutures as we're doing for the wild type. And for the Sutures of the twist 1 mouse, we're just doing interfrontals as our first year goal, and then we'll be doing the coronals after that. Actually, this is bigger than I thought, I guess. So this is the last slide. This is just showing what we'll be delivering to FaceBase. It's basically the gene expression files and associated metadata with the goal that users will be able to look for differential expression, differential isoform use, Go enrichment, and co-expression network analysis, or whatever else you'd like to be using it for. All right, thank you. Are there any questions?
For your twist 1 mutation mouse model, for the coronal sutures, do they get bilateral craniosynostosis 100%?
I'm not sure if it would be 100%, but it's fairly high. It's a high percentage.
And would you consider doing sidedness, like left and right?
Well, I've never seen anything that says like one side or the other is affected, so I don't know if it's a stochastic thing. So we don't have a plan. And also, it would be hard to know at that age, because basically, like once the suture's fused, there's nothing to dissect. But in the coronal suture, if you go through it carefully, the fusion usually starts at one point and it spreads like usually anteriorly or superiorly, a little bit anteriorly or inferiorly. So generally, you'd be looking at it in areas above the fusion, and with the idea that hopefully you'll see any preemptive changes before like outright fusion happens.
So I wonder, for some of your early stage samples, so like E14.5, the gap is pretty big.
Well, E14.5 will only do the coronal suture.
So
I don't know if I could
what
go
kind
back.
of variation would you anticipate, like in your sample?
I mean, I couldn't quantify the variations. So like I said, for E14.5, we're only looking at the coronal. And I've done a lot of sectioning of that for apits. And basically, as you go inferior, or superiorly, it sort of widens out because they're sort of curving away from each other as they form. So we're not really too worried about like necessarily the distance, because I don't think that'll affect the gene expression as long as they're in like within the same age. So at E14.5, we're paying attention to like the telar stage. 16.5 and 18.5, they're much more closer in general, especially the facial sutures. It's the midline sutures that are things that like are the most wide. Palatine sutures, again, like in the body of it, they're closer. Like the extensions of them haven't really started to form very strongly at E16.5, but you still have the bodies that are close together, so there's a definable suture. And like when we've looked at expression of, say, BC11B, you can see that distinction of suture mesenchyme expressing BC11B versus the osteogenic fronts, which are not, or the alkaline phosphatase differential.
Yeah, so if you look at the coronal suture, at the early, like E14.5, you only have the primordium on the side, right? And there's a pretty wide gap on the column.
Yeah, but so the coronal, we're looking at between the frontal and the prior.
Right, yeah.
So that's why we're not looking at midline sutures at that age, because they're
But
too
when
far
you
apart.
take coronal, do you take the top part, or you only take the part?
No, so with the alkaline phosphatase staining, that'll guide us to where we go, because you'll see it. The osteogenic fronts will be close to each other, and then as you go superiorly, those will start to drop away from each other. So we'll just be choosing areas that are close enough to be like a suture. If there are no more questions, thank you very much.